All cellular chemical reactions are going to be catalyzed by enzymes. Enzymes, remember, those are those proteins that speed up chemical reactions without being consumed as part of the reaction. So how does that work then? How do cells use enzymes, and how do enzymes function to speed up chemical reactions? So we talk about metabolism, the sum of all the chemical reactions happening in a cell. That can seem like there's a lot going on, right? And there's a lot going on. There's a lot of chemical reactions building up and breaking down cells. But the key to remember here is that even though we might have this big chemical reaction of glucose and oxygen getting broken down into carbon dioxide and water and producing energy in the process, that process of cellular respiration, that process in all chemical reactions within cells happen in smaller steps that in sum give us a larger overall chemical reaction. Okay, so if we start with a substrate, let's say it's a molecule, it's a molecule, right? That substrate will be acted on by one enzyme, and then it will release the products. The products that are released by that first enzyme are then the substrate for a second enzyme, which changes that substrate just a little bit more, changes it here from B to C, right? So maybe we add on a phosphate group, or we take out a hydroxyl group, or maybe we bond another carbon to it, right? And then that enzyme releases its products, and those products are now the substrate for another enzyme, and so on and so forth. So we're going to talk about the process of cellular respiration, but the key to remember is that each step that we talk about is going to have many enzymes that have just a small part that they play within that process of breaking down glucose to make energy. And this is true for most of the reactions within cells. They're all going to be a chain of enzymes all working together to get to the final product. So we talked about catabolic and anabolic reactions, right? All of those reactions are catalyzed by enzymes. So enzymes can be used to release energy from reactants by breaking apart the bonds in that molecule, right? Catabolism. Or other enzymes can be used in combination with energy and input of energy in order to do work within the cell. Okay, that anabolism or anabolic reactions, right? Usually in an endergonic reaction, those anabolic reactions, energy is required, right? So endergonic, we got to put energy in, and that energy usually comes by breaking up ATP into ADP, right? As opposed to catabolic reactions, we can take energy out, release energy from the molecules, which means sometimes we can capture that energy in the form of ATP. But enzymes can only catalyze chemical reactions that would normally occur, okay? That would naturally occur if enough energy is available, okay? So in order to get molecules to react with each other, like say here we want to switch up the bonds, right? We want to do a recombination reaction, right? Instead of A being bonded to B, we want to bond A to C. Enzymes could only catalyze this reaction if it would happen naturally. And the way it would happen naturally is that these molecules would absorb energy from their surroundings, usually in the form of heat. Okay, thermal energy. Thermal energy causes those atoms in that molecule to vibrate, which can actually, enough heat, those molecules are vibrating, moving enough, those atoms would separate. That covalent bond would break. And then those atoms, once they've been separated, could bond with the other atoms, right? So if we vibrate the bond between A and B enough, add enough energy, heat energy, add enough heat energy to break up that bond, that could then trigger A to bond with C, okay? So that usually takes a lot of heat though, and this much heat, often, that's required as an input of energy to get this reaction to happen, this much heat would kill living organisms, usually, right? Because it would denature their proteins, okay? So what enzymes do is enzymes just lower that activation energy, right? So they lower the activation energy, which means that that chemical reaction is more likely to happen, which also means it happens faster, right? So the input of energy that's necessary for reactions to happen with enzymes present is much lower. We don't need as much energy to get those chemical reactions to happen. But how do they do that? How do enzymes speed up chemical reactions, right? It's their structure, their physical and chemical structure lets them speed up chemical reactions. Okay, especially the structure of the active site, right? So remember the active site, that's where the substrate bonds, okay? That active site has a very specific chemical reaction that will hold that substrate in just the right orientation that will facilitate the change that needs to happen, that chemical reaction that needs to happen to that substrate. So enzymes, once they're bonded to their substrate, what happens is we say it's induced fit. Kind of will wrap around that little substrate like a little glove almost, right? So the process of the substrate bonding to the enzyme causes a slight shape change to that enzyme. And once that enzyme has changed shape, that induced fit around its substrate, that puts everything right in the correct orientation, that now the bonds within that substrate molecule will be stressed, right? So maybe it'll bend that substrate molecule a little bit, so that way those atoms that are in that molecule won't want to bond with each other anymore. It's going to kind of separate them a little bit. Or maybe if it's building up a molecule, adding something onto that molecule, it's going to take two substrates and kind of push them together, orient them so that way their functional groups are close together and it could facilitate that bonding. Okay, so it actually, the active site of the enzyme causes either physical strain on the bonds within that molecule or pushes two substrates together so that way their functional groups can bond together more easily. So let's put it all together. How does this work, right? So substrates are going to bond to that enzyme. Again, this is just random. Everything in the cell is just zooming around, bouncing around each other, that Brownian motion of everything moving around in the cell. Those substrates bond to that enzyme when they just happen to run into that enzyme in the right place, okay? But once they're in the active site, that enzyme, remember that induced fit, it changes shape just a little bit, kind of grabs onto that substrate, and that process of that induced fit grabbing on that substrate lowers the activation energy necessary for that reaction to occur because it's putting stress on those bonds or it's pushing those molecules together to facilitate the likelihood that that reaction will happen. Those substrates are converted to their products, okay? The chemical reaction is carried out, and then their shape has changed. The substrate has changed shapes, so now it's the products and no longer bonds to that active site of that enzyme. Now the active site is ready again, and you can start the whole thing over. So this enzyme can keep working like this, keep going like this until it becomes denatured, right? Until its shape changes, until something happens to its active site. So really the key to why enzymes are able to function properly is their active site, okay? It orients the substrates correctly, it strains the bonds in that substrate, provides a favorable microenvironment, maybe regions of the substrate that are basic, it's connected to parts of the enzyme that are acidic, right? It can also covalently bond to the substrate if necessary, based on the type of reaction that is happening, if that facilitates the process of lowering the activation energy. So enzymes are awesome. Enzymes are great. They catalyze chemical reactions, they keep cells alive, they build up and break down molecules, they help us to release energy, they help us store energy, right? But do we want all of our enzymes working all the time? For instance, we haven't covered this yet, but during cell division, the nucleus, that nuclear membrane that surrounds the DNA, falls apart, gets broken down. And there's an enzyme that does that, right? So the enzyme that breaks apart the nuclear membrane, do we want it working all the time? No, because we'd have to constantly rebuild the nuclear membrane every time we try to break it down, right? So we don't necessarily want all enzymes working all the time. We need to be able to regulate those enzymes. They need some rules to follow, right? So enzymes are regulated. They can be inhibited or activated. So that way they're not working all the time. They need an extra step sometimes so that way they will work properly. So enzyme inhibition prevents those enzymes from functioning, or we can require enzymes to be activated in order to function. But both of these ideas work with we're going to bind something else to the enzyme that will either prevent it from binding to a substrate, or that enzyme cannot bind to its substrate until a molecule has been bonded to it. So in enzyme inhibition, we kind of have two types, but the big idea here is that something is preventing that substrate from bonding to the active site, okay? So in competitive inhibition, we have a molecule that's actually sitting in the active site of the enzyme, but it's not the substrate, and that physically blocks the substrate from bonding to that active site, okay? So competitive inhibition there's an inhibitor molecule that sits in the active site that blocks it from binding to its substrate. In non-competitive inhibition, it's also called allosteric inhibition. A inhibitor molecule binds somewhere else on the enzyme, but not the active site. But in the process of binding somewhere else on the enzyme that we call the allosteric site, that's the location that this non-competitive inhibitor will bind to, the allosteric site. That non-competitive inhibitor bonding to that allosteric site causes the enzyme to change shape. Remember, whenever proteins bind to something, they change shape, okay? That shape change that's caused by that non-competitive inhibitor binding to that allosteric site means that now the active site has changed shape. So now this active site is no longer functional for that substrate to bond to. Okay. Either way though, these are molecules that will bond to the enzyme to prevent it from functioning. These don't always have to be permanent blocks to that enzyme working, right? There are other enzymes that can take off those inhibitors, or at certain concentrations of substrate, sometimes those inhibitors will fall off, right? So these inhibitors can be added on and off. They can be added to the enzyme, they can be taken back off, depending on what's going on in the cell, depending on when they need that enzyme and maybe the concentration of the substrate, things like that. In enzyme inactivation, the proper conformation of that active site is only achieved after an activator molecule has bonded to the enzyme. Okay, so usually activators bond to what we call an allosteric site. Allo meaning other, steric basically meaning location. That once that activator molecule bonds to that allosteric site, now the active site takes its correct shape and it can bond to its substrate. Okay, so we can have enzymes floating around that will only be active once this activator comes in. Sometimes this activator is a signaling molecule, right? Maybe it's a hormone that got sent in from another cell. Sometimes this activator is a substrate that another enzyme produced, and we only need this enzyme to work when that substrate is present. Okay, so activator molecules can be lots of different types of molecules, but the key is they will only turn on that enzyme basically once it's bonded to the enzyme. Either way though, the whole point is we don't want all enzymes working all the time. And guess what? A lot of drugs, a lot of pharmaceuticals that we've produced to treat different things like ibuprofen, Tylenol, basically a lot. I didn't even think of all the drugs I know, but a lot of the medications that we've produced are enzyme activators or enzyme inhibitors, okay? Like beta blockers. Beta blockers help prevent adrenaline from bonding to cells, right? Which helps keep our heart rate low, which helps keeps our blood pressure low. So we can activate, we can inhibit enzymes, but sometimes enzymes need a little help to function properly. So enzymes will often function more efficiently with cofactors and coenzymes. Okay, so cofactors are inorganic molecules, usually metal ions, often zinc, iron, magnesium, things like that. And coenzymes are organic, often vitamins, like vitamin C is a coenzyme that helps to build up collagen molecules in the extracellular matrix, right? So vitamin C is a coenzyme that helps the enzyme itself that builds collagen. Vitamin C helps that enzyme function properly, okay? So another example of a cofactor is like the iron that's in the heme group of hemoglobin. This iron ion right here is really how hemoglobin is able to carry oxygen, right? Because this iron will have a +2 charge, oxygen has a -2 charge. Now this iron's gonna grab onto the oxygen, right? So it's an inorganic substance that helps that protein or that enzyme function properly. So these are they're not truly activators per se, but the presence of these inorganic and organic cofactors and coenzymes help enzymes function more efficiently. Sometimes the products of an enzyme pathway, of a metabolic pathway, can also inhibit enzymes, right? So here we have this enzyme, it's gonna break down this substrate, right? It's gonna change it from a triangle to a rectangle. Now we're gonna add some bumps on this rectangle by another enzyme, and then this enzyme changes this bumpy rectangle into a square, right? Point being, it's a chain reaction of multiple enzymes working together. In the process of what we call feedback inhibition, the final product of this enzymatic pathway can bond to this first enzyme and prevent it from now bonding to its substrate. So basically we're saying once we get enough of this product, of our final product, we don't need to make any more. We don't want this reaction to happen once this concentration reaches a high enough level. And so by this end product actually being an inhibitor of the first enzyme in this pathway, it shuts the whole thing down. Okay, so this is how we prevent getting too much of a final product. We can inhibit enzymes along the way. And feedback inhibition. So the reason we talked about enzymes first is because as we talk about cellular respiration, as we talk about anything that's going on in a cell, it's going to be catalyzed by enzymes. Okay, in our class, we won't specifically learn each and every enzyme along the way. Okay, I'm not expecting that we know the names of all the enzymes for every single step and what they produce in cellular respiration, but I want us to understand that this happens in many small steps, and each step is catalyzed by an enzyme. And we can turn off, we can turn on, we can inhibit those enzymes in many different ways.